Good morning, everybody. Welcome back to day 2 of the Rare Patient Advocacy Summit. Thanks for joining us today in track 4, Science, Technology, and Innovation, sponsored by PRA Health Sciences. You can go ahead and download the app if you guys haven't already. I'm Jessica Shiles, and I will be track host for this morning's sessions. I would like to also welcome those of you joining us remotely through live stream. I would like to remind everyone to, again, go ahead and download the app that we have in order to keep up to date on some of our highlighted features, like the live Q&A. Today, we will be joined by Shaun from Monahan, Family Associate Professors of Rare and Neglected Diseases at the University of Notre Dame, as well as Wes Miller, Medical Director at Sangamo Therapeutics, who we will also be diving into a discussion on new approaches that are being developed to address the cause of rare diseases during our sessions of rare therapies, pharmacological chaperones, and cell therapies. Let's welcome them up. Well, they're already here. And this is Shaun.
That's the clicker. Great. Can you hear me okay? Yes. Great. Well, thank you very much. I'd like to thank the organizers for such a wonderful conference and for allowing me to come here and present our work to you today. So let's dive into this. So I'm going to talk about how we got started in looking at how genetic mutations affect protein misfolding in rare diseases. So this is the general outline of my talk. I'm going to give you an introduction into what happens when proteins misfold and why genetic mutations cause those kinds of defects in protein misfolding. And then I'll tell you a little bit about our work on looking at a rare disease called Sanfilippo syndrome and the enzyme that's involved in the defect that is responsible for Sanfilippo syndrome, and how we got involved in looking at these genetic mutations in the syndrome and coming up with an algorithm to better understand how genetic mutations would affect protein folding in such a rare disease. So I want to start with a very basic introduction, and we all know that proteins are the building blocks of life. So when you're talking about very basic things like actin, these things that build cell architecture, or enzymes that do very basic functional processes in the host cell, right? Or hormones that affect communication, like growth factors, these are all proteins. And so proteins are the essential building blocks of our cells. And, sorry, can I back up? Let me see if I can. Is there a way to push play on that? Probably. So what's supposed to happen is that thread is supposed to wind itself around and turn itself, oh, there it goes. Okay, it went on one of the slides. Sorry about that. So every protein starts out as a linear thread, and it goes through a series of folding parameters, and then it finally adopts its native fold. And the way to understand this is that proteins that start out as these linear threads must fold into a 3D structure properly in order to function properly. So you can imagine as it like origami. So if you have a sheet of paper, which is your thread, if you fold that sheet of paper properly, you get this nice, beautiful crane, and it goes on in the cell to do the work it needs to do. But if you can't fold it properly, and then what happens is the cell actually has a mechanism to recognize that that protein isn't folded properly, and it puts it into the recycle bin. And there is actually a cellular structure called the proteasome, which is responsible for degrading unfolded proteins. One of the things to appreciate is that protein folding is really a complex puzzle. If you take a very simple protein that has, let's say, 27 little tiny subunits, it starts out as over 10 to the 16th power, billions and billions of possible ways that this little thread can adopt its three-dimensional fold. But there's really only one best state that is responsible for making that protein fold and function properly. So it's really a complex puzzle, and it takes a lot of intricacy by the cell in order for the protein to fold properly. If we look at why proteins misfold and how genetic mutations are linked to such things, here's a typical example where a single point genetic mutation changes the amino acids or the building blocks of those proteins from something that looks like something very simple like a valine, for example, and just a single nucleotide change will then tell the protein to put a different amino acid there, and you replace that with something like a tryptophan. And when you put that kind of big structure there where it shouldn't be, it really almost creates a knot or a kink in that thread, and that's what really causes a lot of these defects in protein misfolding. So what happens when these things happen? Here are some of the consequences of protein misfolding. The protein is either rapidly degraded, and so what you have is the pools of functional protein are really not made because it keeps going into the trash can. The protein can have structure, but a little bit of improper folding will prevent protein function. So you may get the presence of slightly misfolded protein. It may accrue, but it's not really perfect at doing its job. And finally, if it's really bad, these proteins can accumulate and aggregate and really clump together and cause serious problems for the host cell. If you think about many of the major diseases like Alzheimer's and Parkinson's, these are really the consequences of proteins not being able to fold and behave properly, so they aggregate very quickly and form these huge insoluble aggregates in things like neurons, resulting in cytotoxicity. But not only common diseases, I want to point out that rare diseases, many of the ones that you know about, are really due to defects in protein folding. And what you can appreciate in these pictures is really that if you look at a given rare disease like Fabry or Gaucher, you can see just by looking at the protein structure that it involves many, many intricate folds. And there's a lot of work involved in making this into a proper folded protein. So with that, I want to introduce a little bit of our work on this rare disease called Mucopolysaccharidosis. And it's essentially a lysosomal storage disease. And simply put, what happens is there's a cascade of enzymes that work to break down these branched sugars inside the cell. And when one of these enzymes don't work, the branched sugar cannot be broken down, and the sugar causes toxicity buildup in the host cell. So MPS IIIA is caused by, there's 4 types of MPS IIIA, and what we're looking at is MPS IIIA, which is caused by defects in this one enzyme called sulfoglucosamine sulfohydrolase. And that's essentially one of the enzymes that acts to break down these sugars. This enzyme is located in a specific compartment of the host cell. It's an autosomal recessive disease. In terms of clinical symptoms, the patients usually begin in childhood and have severe neurological defects, dementia, aggressive behaviors, hyperactivity, seizures, and loss of vision. There's really no specific treatment for this particular one at this time, and a lot of times the affected people don't live past the 2nd decade of life. There have been strategies to try to treat things like mucopolysaccharidosis. One of the more common ones is enzyme replacement therapy, and I think many of you are familiar with enzyme replacement therapy approaches, and it's really replacing the defective version of that enzyme and adding in or treating by the addition of a properly functioning enzyme so that it can break down the sugars. The limitations with this include insufficient biodistribution of the recombinant enzyme. You need to get it into the cells that actually start building the toxicity up, especially in the case of neurons. That's difficult to do, and the high costs involved in doing enzyme replacement therapies. So an emerging technology has been to use what's called pharmacological chaperone therapy. And this has been really heavily investigated, especially in rare diseases where we think protein misfolding constitutes a lot of the cause behind the rare disease. One of the things to point out is that the approach in using chaperone therapies can only be applied to patients carrying chaperone responsive mutations. In other words, if you have an enzyme where the catalytic or the functional domain doesn't work, no matter how you get that protein to fold properly, if the catalytic site doesn't really work, then the enzyme won't function. I want to just give you a little bit of an introduction into chaperones. So although we talk about pharmacological chaperones, our cells actually make what are called chaperones. And these do exactly what we think they do, is that they help protein threads form their three-dimensional structures properly. And they give it a little bit of a helping hand. And the best analogy I could come up with is this very outdated picture of an iPhone at this point. But what you see in that cord are these little plastic things. And they're basically put there to keep the cord from tangling. And I think all of us have experienced that. And these are essentially what chaperones do. They give your protein thread a little bit more time so that it doesn't clump up. All right, so this is really an attractive way to treat many of these protein misfolding diseases. And this is just a quick slide showing you that many companies are interested in pharmacological chaperone therapies. Some of the limitations into the possible use of pharmacological chaperones, the main one seems to be that there's a large heterogeneity of mutations in the patient database. Right? Not all mutations may be responsive to protein chaperone therapies, but we don't know which ones can be and which ones not until we maybe treat the patient. And secondly, there's really no definitive assay to test for, in the case of what we're looking at, MPS IIIA. How can we understand whether if we apply a chaperone therapy, especially in a cell-based model, whether that's going to actually help the protein fold and function properly? So we ran into that limitation when we were looking at Sanfilippo syndrome. The other thing to note is that when we looked at MPS IIIA, we realized that there were over 100 patient mutations in this disease. And so the first question really became, how can we predict how these individual genetic mutations contribute to the ability of the protein to misfold, giving you that example prior where a single point mutation will cause a specific change in the amino acid thread. So then we came up with this idea that maybe what we could do is use computational tools to really figure out whether that single genetic mutation will cause a change in that amino acid and how that change in the amino acid will really affect the ability of that protein to misfold. So that was our multi-parametric computational algorithm, and I know that sounds very complicated, but I'd like to simplify it by saying that this is essentially a survey that we're giving to the enzyme to ask, can you fold properly? So if you ask it a series of questions, will my genetic mutation slow down my protein thread production? Will my mutation generate these little knots in the protein thread? And so just to give you some examples, these are biophysical and biochemical parameters, but we can simplify it in terms of questions like this. Do any of your relatives or enzymes that look like you in non-human hosts have this mutation? Yes or no? If it's a yes, then it's a point. If it's a no, it's simply not a point. Does this mutation help certain oily parts of the enzyme to try to mix with water? Yes or no? Is the new mutation giving you an amino acid now that's too bulky, like that example that I showed you, to now fit where it should be in the pocket of the enzyme? Yes or no? And we give it a score, and so on and so on. Could the mutation interfere with the cellular control over where and when in terms of its location in the cell? And so we actually did this for all of the mutations that were available in the database of MPS IIIA at the time. These are all patient mutations that have been observed, and we scored all of them. And those are the parameters that you see on the left, things like translation rate, aggregation propensity, et cetera, et cetera. And so what we did with this algorithm was to ask then, will our algorithm really correlate with some of the symptoms of the disease? And in many cases, we found this to be valid. So we're really using predictive algorithms. In other words, just computational tools, no clinical testing, no real biological data. And in many cases, we find that the higher the mutation score in our algorithm, the more severe the disease and the earlier age of onset, telling us that maybe this is a way that we can almost predict the severity of the protein misfolding when it comes to looking at a single genetic mutation. So that was our approach, and that was our publication, and I just want to highlight the points. This analysis shows that many of these disease phenotypes, especially for this one disease, is specifically due to defects in protein misfolding. None of the mutations that we surveyed actually destroyed the catalytic activity of the enzyme. They were all in other places that affected the ability of the protein to fold properly. These scores provide a guide for which patients could be treated, perhaps using chaperone therapies, and the algorithm advances a key understanding and the biochemical understanding of this rare disease. And we think that's important for a rare disease where there really isn't a ton of data regarding mutations. Many of the proteins have not undergone extensive biological testing. So we think this is a great first foray into understanding how these individual genetic point mutations correlate with the actual biological data in terms of protein misfolding and protein misfunction. And the design of the algorithm can be tailored not only to Sanfilippo syndrome, but many of the parameters are not unique to a particular enzyme. But we do tailor every multi-parametric algorithm so that we can add in additional parameters that are specific for the enzyme or the disease of interest. So finally, I just want to show you some of our actual biological work. And what we did when we scored these mutations is to look at one that we thought had a pretty significant score and say, well, if this has a pretty good score on our algorithm, the protein should misfold. And when we actually put it into a biological system like a cell, we should be able to see that. And so that's some of the work that we're doing now, which is to better understand the cellular mechanism by which many of these proteins misfold, and also to establish a biological assay so that we may be able to test if we have any of these chaperones. So here's just a clear example, and if you've not seen cell culture, what you can clearly see is that in the upper panels, there's a lot of green, and that's the healthy cell producing the functional version of this enzyme. And in the bottom panels are the cells that have the mutation in this enzyme, and what you can see is they are not being really properly made in these cells. So the idea is to use these cells as a system to be able to say, well, if we have the absence of that green normal enzyme and we add a chaperone, can we rescue the presence of that functional enzyme using this kind of fluorescent readout? So ultimately, we envision our pipeline to be included in the following. One of the things that we can really do is to use this algorithm and say, predictively, what are the mutations in the patient database that are likely to be more severe, less severe? Maybe there's a subset there that might be more amenable to chaperone therapies. And then that can guide, these mutations can help us guide. The limitation is that we can't make all 100 or 150 mutations in many cases in biological systems. So if we are to build representative cell lines or animal models, which are the mutations that we will choose that will best appropriate protein misfolding that can be treated with things like pharmacological chaperones? And finally, one of the things that we hope is that once we refine these algorithms, they can be a way to guide whether certain patients that have genetic mutations when they're tested can be amenable to chaperone therapies. I just want to conclude by acknowledging all of the people. This started with a really wonderful postdoc in our lab who had a biophysics PhD, and that's Krasyu Ugranov, along with many of my students at the University of Notre Dame, where I am. And we have Rare Disease Day events at Notre Dame every year, and Dr. Kasturi Halder, our director of the Böhler-Perseguian Center for Rare and Neglected Diseases, has sort of modified our Notre Dame fight song, if you're familiar with that at all. And she says, "What though odds be great or small, old Notre Dame will win over all, while her loyal students march in the fight against rare disease." She's very proud of that little bit of modification. So finally, I'd like to acknowledge all the people that I'm affiliated with, especially Dr. Halder at the Böhler-Perseguian Center for Rare and Neglected Diseases, Dr. Richard Taylor and Brian Blagg at the Warren Family Research Center for Drug Discovery, my lab and all the members, support from the NIH, and finally the Monahan family for support of our work. Thank you very much. Okay.
All right, I think we're going to do questions at the end. Is that correct? Very good. Thank you, Shaun. That was very interesting, and I'm looking forward to asking you a couple of questions about that data. So my name is Wes Miller, and I'm a medical director at Sangamo Therapeutics. We are a small biotech company, but growing biotech company up in the San Francisco Bay Area. And it's an absolute privilege to be here at the Global Genes Rare Patient Advocacy Summit. So I am a pediatrician, and I joined the Sangamo team up near San Francisco very recently. And prior to that, for 12 years, I practiced as a pediatric hematologist and oncologist. And as my time on faculty at the University of Minnesota, my clinical and research interests and foci were on the use of allogeneic hematopoietic stem cell transplantation and other cell therapies for children and adolescents and adults who have rare inherited metabolic disorders. So the rare disease space is very near and dear to my heart as a practicing clinician. In many ways, BMT, which is that therapy, allogeneic stem cell transplant, laid a lot of the groundwork for a revolution that we're seeing in modern medicine, which is the advent of genomic medicines or gene-based treatment strategies. When BMT started back in the late 60s and early 70s, some of the initial patients treated were young children who had life-threatening monogenic disorders such as severe combined immune deficiency syndrome or SCID, as well as osteopetrosis. And in a very crude way, successful allogeneic BMT showed a proof of concept that if you transferred normal, healthy blood and immune systems carrying good copies of specific genes into these young children who had a faulty genetic disposition, that you could actually significantly benefit that child or that adolescent or young adult. And that was important information. But as a bone marrow transplant doctor, it's also important for me to tell you that that is an incredibly risky and barbaric therapy. And it's not elegant at all. I think of it very much like a bull in a china shop. And while it has its place, and certainly in the last 4 decades it's become better and slightly more elegant, it leaves a lot to be desired. And so the advent, again, of genomic medicines has used that experience and brought to the table much more specific and elegant ideas about how we can start to approach treating in a meaningful way patients who have rare diseases. So I just want to say before I launch into this, though, that I trained for at least 11 years to become, I have to think about that, to become a pediatric bone marrow transplant doctor. And near the end of my time as a practicing clinician, I was participating as an investigator on numerous gene-modified cell therapy trials, which was so awesome and incredible to see for our patients and our families. But even with all my training, I had a difficult time wrapping my brain around the emerging genome modification and genomic medicine treatments and technology that was coming out there. And I saw that struggle in my patients and families as well. So really the goal today is to try and give you an overall framework and a general understanding of these various tools that are now starting to populate this growing and expanding toolbox of gene-based treatment strategies. So before we talk about those specific tools, I think it stands to reason that we should take a step back and say, why genomic medicine? So why is there this revolution that's happening in the landscape of modern medicine? And I can think of several good reasons why, and I'm sure there are many more. So one good reason is that, of course, we now know that much of human disease is rooted in faulty genes. And as Shaun was talking about earlier, genes are the coding units within our DNA that serve as blueprints and instruct our cells on how to make structural and enzymatic proteins, right? And our cells have to make normal functional copy of those proteins in order for the cell to be healthy and to function, which then allows the organ and the tissue to work and then leads to an overall healthy body. And so if any of those genes is faulty, then there are some potential significant and serious downstream consequences. And obviously, the easiest way to conceptualize this is to think about well-established monogenic disorders. So these are diseases that are well known to be mapped to a particular gene that is faulty. And examples of which we've heard today, MPS IIIA, which is one of the Mucopolysaccharidoses. There's multiple other lysosomal storage diseases out there that involve a dysfunctional single enzymatic protein that leads to a disease state. So the gangliosidosis and the leukodystrophies, the glycoproteinoses. There are some slightly more common disorders that we're all, I think, familiar with, like the hemophilia disorders, hemophilia A and B, which are defects in the genes that code for important clotting factor, enzymatic clotting factor proteins, so that the blood can clot and achieve hemostasis when it needs to. I think a lot of us are familiar with various forms of hemoglobinopathies, so patients who are born with mutations in the genes that code for the proteins that are subunits of hemoglobin, which is the most important protein within our red blood cells. Diseases like sickle cell disease, beta thalassemia, alpha thalassemia. As I mentioned earlier, diseases like severe combined immune deficiency syndrome. So there's many, many examples out there of diseases, so-called monogenic diseases, where a single genetic lesion or abnormality maps to a clear disease state. The second reason that I can think of is that medical science has also provided a groundwork and a roadmap for the notion that you can correct these genetic disorders by simply providing normal functional protein. So if we go back to these same diseases that I was just mentioning, for instance, hemophilia, for a long time now, we as hematologists have been able to treat patients with hemophilia using recombinant enzyme replacement. That is, we have a synthetic form of their missing enzyme, which is their protein that they can't make. We can infuse that to the patient, either on demand or prophylactically, to help to prevent life-threatening bleeding episodes. Similarly, for patients with hemoglobinopathies, protein replacement exists in a crude way with red cell transfusions. We can transfuse patients who have sickle cell disease when they need it, or thalassemia syndromes when they need it. Similar examples exist as well, as Shaun mentioned, for some of the lysosomal storage disorders with enzyme replacement therapy. So to be clear, we know with this roadmap that if you can provide the functional protein, you can have therapeutic benefit. And we gotta acknowledge that there has been a significant improvement, therapeutic improvement for patients with some of these disorders with protein replacement therapy. But one of the limitations of that approach is it is not permanent, right? It's not going to the blueprint and saying to the cell, each and every cell, hey, let me correct your underlying code problem or your blueprint problem. It's sort of like a Band-Aid. You're constantly having to give the protein time and time again. And so there are some limitations about that approach. And then the third reason why I think we're seeing an advent and an explosion of all of these tools within our gene-based treatment strategy toolbox is because the technology is now here, it's arriving. And so to be clear, a lot of the early molecular work that went on to understand how to change DNA or change the genome started 2+ decades ago. But it's only recently now that our friends who do this work in the lab have been able to really understand how to make true tools that we can translate into the clinic for therapeutic benefit. And so with all of these growing tools in the toolbox, again, the vernacular and the terminology gets a little bit confusing at times. And so what I hope to address in the remaining time are 5 of the most common questions we hear at our biotech company, which has a focus on gene-modifying therapies. And that is, what is gene therapy? What is genome editing? How are those similar? How are they different? What is cell therapy? And how is that similar or different to gene therapy and genome editing? Can these various tools be combined? That is, if you're working on a project, can you pull out both a hammer and a screwdriver from the toolbox? Well, yeah, the answer is you can. Depends on what the underlying project is or what that disease is. And then, of course, what does this mean for the rare disease community? So I'm going to start with gene therapy. That's probably the most recognizable treatment approach and tool in this toolbox. Gene therapy in its simplest form says the following. We know that we have a faulty gene. Therefore, let's take a good copy of the gene and insert it into the cell, tissues, organs, body habitus of that person affected by that genetic disorder. All right, that sounds very simple, but those of you who know a lot about gene therapy know it is not simple at all. And there are numerous hurdles to actually operationalizing this concept and this idea and getting this to work within real people. So the first hurdle that we have to think about is, what cells do we want to correct, right? Because if we want to correct every cell in the body, that's a tall order. We might have a limited number of good copies of the gene that we are trying to transfer into our patient. Where do we want to correct these cells? Do we want to correct them in vivo? That is, do we want to insert the good copies of the gene into the body and have them introduce themselves into the cells? Or do we want to remove cells from the patient outside of their body and in the laboratory perform the gene transfer or the gene therapy? So the former is called in vivo gene therapy, and the latter is called ex vivo gene therapy. And as you can imagine, there may be some advantages to each of these and disadvantages to each of these. A particular advantage of ex vivo gene therapy is that we end up with a more limited finite number of cells that we're trying to correct, which again operationally may be a bit easier than trying to correct orders of magnitude greater numbers of cells in vivo. But the next question is, how do you actually get a good copy of a gene into a cell that needs it, right? And the answer to that is we have to use something that's called a vector. A vector is either a living thing like a virus or a non-living thing like a particle that can carry a good copy of a gene across the cell membrane into the cytosol, or the cytoplasma of the cell, and then furthermore into the nucleus. And then that vector may or may not be able to take that good copy of the gene and integrate it into the DNA that already exists. Why is that important? If it doesn't integrate that good copy of the gene, the therapeutic copy that you've inserted, then if and when that cell decides to divide and make two daughter cells, it's not able to give one good copy of that transferred gene to each of the daughter cells. However, if the technology you're using allows that therapeutic external gene to integrate into the DNA, then it can pass a good copy of that gene onto each of its daughter cells. Those daughter cells, if they divide and make new cells down the road, can also pass on good copies of those genes. So you can see that there might be in some diseases and in some cell populations a need or an advantage to performing gene therapy with a particular technology that allows for integration of the gene into the DNA. Those are but two concepts or two considerations when we're thinking about gene therapy. There are lots of other things to worry about and hurdles to overcome. So one particular hurdle is if you are using a vector that is in fact a living thing like a virus, if your patient has been exposed to that particular virus just in the course of normal living, right, then they might already have an immunologic memory to that particular virus that's now trying to carry the good copy of your gene or that gene of interest into your patient's cells and patient's body. They therefore might reject or fight off that vector, and if they do that, then it may not live to see the day that it's able to actually carry the good copy of the gene into the cells of interest. Make sense? Furthermore, the field is continuing to try to overcome the notion that if you try to do in vivo gene therapy, you may need or want to direct your good copies of genes to certain tissue types of interest. So a lot of work is ongoing in creating what are called tropic or tissue-specific vectors that are designed to only copy, let's say, good copy or carry good copies of genes into nerve cells or neurons, as we heard about earlier with SMA type 1 gene therapy, or maybe to try to only carry those good copies of genes into liver cells or to lung cells, or whatever the case may be. So I think of gene therapy, that first tool in the toolbox, a little bit like a crude tool. I think of it like a hammer. It's just trying to take good copies of DNA, of genes, and hammer it into cells. It's not very precise. It does its best to get it into the cells that we want to get it into. It might succeed, it might not. And when it does succeed, it might carry many copies of a gene, a good gene, into one cell, whereas normally we might only have, say, 2 copies of that particular gene. And that may or may not be a problem. It may be a good thing if there's more of that gene per cell. But if gene therapy is like a hammer, The next tool or group of tools we're going to talk about, which is genome editing, is more like scissors. And I think of these particular tools, this strategy, as molecular scissors. So genome editing is a related group of technologies or treatment approaches that take molecules, introduce them into cells of interest that you want to modify genetically, and then directs those molecules specifically to a part of the DNA, only a single place if possible in the DNA of interest, and then cleaves that exact location within the DNA. So in contrast, when we talked about gene therapy, especially integrating forms of gene therapy, often that's done in a not very specific way, in what we call a random or semi-random way. That is, maybe the viral vector that's carrying the good copy of the gene, it could insert it into a part of the DNA that's nowhere close to where the normal gene that you're trying to correct would live, okay? But genome editing is different. It's more like, I think of it like a word processor, where you can do a Control F to find a specific sequence of DNA that you're interested in. You can then highlight that specific sequence of DNA. That highlighting basically happens based on the specificity of these handles of the molecular scissors that you're putting into the nucleus of your cell. And then you snip with something called nucleases, which are special enzymes that can create double-stranded DNA breaks, completely cut through the entire helix of the DNA. Once that happens, you then rely upon your cell's own existing endogenous DNA repair mechanisms to try to repair the break. When that happens, the cells can do one of several things. First of all, they can get it wrong, which they often do, and then that fundamentally changes what the original part of your DNA was. So if it was incorrect, then you can correct the DNA. If the gene was correct in the first place, you can actually create an intentional mutation. There are some situations where that might actually help a disease state, believe it or not. So these molecular scissors, like this part of our toolbox, is definitely more novel and recent than the concept of gene therapy as a tool. And the three major platforms of genome editing that are emerging and that have emerged are zinc finger nucleases, transcriptor activator-like effector nucleases, or TALEN, and clustered regularly interspaced palindromic repeats, or CRISPR, which then has a CRISPR-associated protein called Cas, which is sort of the blades or the nucleases that cut. All three of these are basically the same concept, which is saying these are molecules or platforms that are able to seek out and find a specific sequence of nucleotides, DNA nucleotides, to localize scissor blades to the gene of interest, and then to create a cut within that gene. When and where these might be applied, and how they might be applied, and whether they might be more advantageous for certain diseases is definitely a topic of hot debate in the medical science community. And it's becoming clearer that there might be certain types of mutations that one wants to correct or create that might better be served by one of these types of scissors, so to speak. So we talked about gene therapy, which is a bit of a crude way of modifying the genome, getting genes into cells in a not very specific way. We talked about genome editing, which is much more specific. And then the last of these tools that I want to talk about is cell therapy. Cell therapy is the tool that I'm most familiar with as a pediatric blood stem cell transplant doctor. And as I said earlier, in its original kind of nascent form, laid a lot of the groundwork for this revolution of gene-based cell treatment strategies that we're now seeing. In its purest form, cell therapy simply means the application, whether it's infusion or otherwise, of cells to a person for a therapeutic effect. So this means that simple things like red blood cell transfusions or platelet blood cell transfusions are forms of cell therapy. It also means that more complicated treatment strategies like allogeneic bone marrow transplant that I talked about earlier is a form of cell therapy. But of course, in the last decade, we've had other emerging forms of cell therapy as well. We've had mesenchymal stem cell therapy or mesenchymal stromal cell therapy, also called MSC therapy. Of course, we've had the advent now of induced pluripotent stem cell therapy, which has lots of promise and is almost certain to have lots of application to treat disease. And then today, we can also deliver other novel forms of more developed cell therapy, including lymphocyte forms of cell therapy. And that's a good segue into asking the question, can all of these different tools from our toolbox be combined? And the answer is a resounding yes. And this is happening. This is happening already. And many of you are already familiar with it. So some classic examples are when we think about the combination of hematopoietic stem cell therapy and both or either gene editing or gene therapy. So there have been numerous trials now looking at the effect that this might have in correcting certain genetic disorders and diseases. Another example is the advent of CAR T therapy, which stands for chimeric antigen receptor T lymphocyte cell therapy. So this is an example of a more developed, not stem cell therapy, but more developed T lymphocyte group from a patient that's taken, population from a patient that's taken out of their body, and then genetically modified to make it more capable of killing off cancer that the patient might have. And of course, we've now know that we're seeing really impressive outcomes in patients with certain types of blood cancers like lymphoma and leukemia. And in fact, as many of you know, CAR T therapy has now been licensed for use in certain indications in the United States. It's also possible that genome editing and gene therapy can be combined in some ways. Already, we know that in the clinic and in trial are gene editing approaches that not only create a very specific, precise cut at a certain location in the DNA, but also provide what's called a donor template, so that the cell, as it's trying to repair that cut, can read through that template, which has the correct sequence, so that it can correct its own previous mutation. So, very, very high-level overview that I wanted to give you today, just about these emerging strategies and what they are and how they might be different. And obviously, you know, the big question that weighs heavily on all of our minds is, what does this mean for the rare disease community? And I think all of us are very hopeful and optimistic that the advent of genomic medicines and gene-based treatment strategies is going to mean a lot for patients who have rare diseases. We're already starting to see that. If you go to clinicaltrials.gov and just search gene editing and gene therapy, you're going to see hundreds and hundreds of trials, whereas only a decade ago, you only saw handfuls of such trials that were in the clinic. A word of caution is that, as you all know in this audience, medical science doesn't always move as fast as we'd like it to, right? And one of the things that I think is important to consider is that there are certain new treatment approaches, such as PCT, like Shaun talked about earlier, which may be able to get through, maybe not, but may be able to get through certain regulatory hurdles a little bit faster as a single molecule that might be applied to various diseases. Versus when we start thinking about gene-based treatment strategies, such as gene editing and gene therapy, we may have to go slower and really do a very rigorous evaluation with respect to safety and efficacy for each particular genetic disorder that we're trying to treat. Almost certainly, because this science is changing so fast, in 3 to 6 months from now, there are going to be new tools in the toolbox or emerging tools in the toolbox. And I think what's going to be key is that experts within each specific genetic disorder link up with experts who are creating these tools to really understand how to best apply them for that particular disorder. And so with that, I'm going to stop. Thank you so much for your attention, and I'm sure Shaun will be willing to entertain any questions. And I am too, as well. There's a question over here. Yep, and then we can repeat it if you'd like. Yeah.
Sure. So my question is twofold. One, are you hiring in the greater DC area?
Sure. I don't know if we're hiring in the greater DC area, but you can go to our website, which is sangamo.com, and maybe sort that out. In all seriousness, my email address is wmiller@sangamo.com. Please feel free, anyone, if you have questions or just want to reach out and get in touch. I'd love to hear from you. So what does this actually look like? And this is where, where does the rubber meet the road? Sort of what is this going to actually mean for patients? So in the current iteration of blood stem cell therapy that's being combined with either gene editing or gene therapy. So you take the patient's own blood stem cells out of their body, and then you either perform gene therapy or gene editing on them, and then you give them back to the patient. In general, the hope is that that's a sort of one and done kind of therapy. And for most of the diseases this is now being used in, that's a reasonable expectation. The problem is, as it currently stands, in order to make space to give these new modified cells advantage, we have to give a fairly significant round of chemotherapy to the patient. The reason we do that is not because they have cancer. We do it to take away their existing blood stem cell population, which has not been genetically corrected, right? It's like uprooting all of the plants in a garden and then replanting it with new seeds, genetically corrected seeds, to give those new seeds a survival advantage so they can repopulate and then provide whatever that missing protein of interest was, if that makes sense. So that's one area where, even though that's a very common approach right now in clinical trials, that's one area where obviously there can be some improvement in medical science because it's not fun to have to get chemotherapy. To be clear, right now, I think for a lot of these diseases, the risk-benefit equation clearly tips to trying these therapies because there's nothing else for them. And so that chemotherapy pill is while it's difficult to swallow, it's acceptable to swallow it, if that makes sense. But that's an example of where we might need to get better. For in vivo gene therapy, the jury is still out on your question because it depends on the types of cells that you're correcting, whether they are stem cell populations that within the body can continue to give rise to daughter cells, which also carry the genetically normal variant that you've inserted or introduced. And it also depends on how good and specific your vector was at getting it to the cells of interest, right? If your genes, your therapeutic genes got diluted out, it might work for a little bit of time, but you might have to repeat it. And then the problem comes in if you're trying to use the same kind of vector, and if that vector was a virus, and if your patient has now developed an immune response to that vector, they may not be able to receive that same exact treatment again. If you want them to get that same gene construct, it may have to be attached to or packaged within a slightly different vector that their immune system has never seen before. Complicated, right? It's very complicated. And I think the best advice is, as these therapies, as gene-based treatment strategies emerge for all of the specific and rare genetic disorders, to really talk with the clinicians who are running these studies to understand what it means for you or your child. Yeah.
Can I ask a follow-up question
Yeah.
for that? Are there special considerations for genetic diseases that affect neurons and whether there's a critical time at which this therapy can be applied there?
Absolutely, right? So as I said, we've learned lots from the application of blood stem cell transplant just in its original form in the 1960s and 70s. But one of the very early things that we learned, even if you just look at a group of related diseases like lysosomal storage disorders, that for some of those diseases, it helped immensely. And for other diseases, it didn't seem to help so much. And a lot of it was probably had to do with when the children were coming to clinical diagnosis and presentation, and how much damage maybe had already happened in key organs that were susceptible to damage from their underlying disease. So, Shaun knows the answer to this quite well with certain types of Sanfilippo syndrome, blood stem cell transplant is not helpful. And so one of the challenges with gene therapy for Sanfilippo and for MPS IIIA, especially the neuronopathic forms of the disease, is how do you get it in early enough, soon enough, and in large enough quantities that you're likely to have an impact on the eventual natural history of the disease.
Great, thank you, Shaun. We also have a question here from our live question and answer that I'm just going to kind of tack onto that. Are any of these gene therapies targeting the germ cell line? Why or why not?
That is a very good question. To my knowledge, germ cell gene therapy is very experimental. I don't know if anyone is actually doing that for targeted purposes right now. I mean, on the flip side, we also know that there are ways in which one can, through things like pre-implantation genetic diagnosis and IVF, sort of have a bit more control over the passage of certain genes or traits onto children. If anyone knows better than I do about germline gene therapy, I'd love to hear from you.
Other questions?
This gentleman may know.
Wes and Shaun, thank you. You know I don't know the answer, Wes. But this is a question for Shaun. The chaperone therapies that you were talking about, can those be applied to the other Sanfilippo types, most specifically type C, where other modalities are not applicable because of a lack of a bystander effect?
Lack of a, sorry, could you
A bystander effect where you can, get efficacy or distribution from cells that are next to one another?
Yeah, that's a good question. I think the PCTs, and I am definitely more of a research scientist, but PCTs in my mind are going to be limited to those where, one, the protein is really almost trying to get there and fold properly, and if you can give it that dose effectively, that's going to help it fold and catalyze properly. We're especially interested in how these algorithms relate to cases where these proteins are part of a larger network of protein-protein interactions, and where you have things like, even if you were to deliver chaperone therapies, maybe we're missing something and the protein can be folded properly, but the genetic mutation is often affecting something else in terms of protein-protein interactions, and we're not addressing that with our algorithm. I think with the bystander effects, definitely, whether you have compensating effects, that's tough to really evaluate in terms of whether doses of protein chaperone therapies are going to be effective, I think. Yeah.
Hi, thank you so much. I've done stem cell therapy myself, and it was life-giving. So I appreciate your presentation. I was wondering if you could explain the difference between mutation and variant.
Sure. Would you like me to take a stab at that?
Sure.
Yeah.
Sure.
Yeah. So I think a variant just means that, in general, I think in genetics, that there is a sequence of DNA within a region of interest, usually a gene, that seems novel or different than what most people have. If that causes disease, then we generally call that a mutation. If it doesn't cause disease, we call it a benign variant or just a polymorphism, maybe a benign polymorphism. If we're not sure, because no one's ever described it before, and it maybe has a chance to cause disease, but we can't be certain, then there are fancy now sort of in silico or computer-generated algorithms that might be able to predict how likely it is to cause disease, that is to be pathogenic. But still often what happens is the geneticists say, this is just a variant of unknown significance or unknown clinical significance, and we sort of have to wait and see. Does that help to address your, yeah. I had a question for Shaun. So one of the things as a clinician that seems really attractive to me about PCT is the concept that, you were talking about chaperone therapy for MPS IIIA, but you can imagine that the same exact chaperone molecules might have a therapeutic benefit for lots of different enzymatic disorders. So how easy is it to predict the applicability of a given chaperone, pharmacologic chaperone, to various disorders? Does that question make sense?
Yeah, that's a great question. One of the things, in my mind, I think of the best, so heat shock proteins, the native cellular version of cellular chaperones, right? So when your temperature gets a little too high in these cells and the proteins start to misfold because their thermodynamics are off, the cell builds up these cellular chaperones to try to help them. And those may be specific and can be general, right? So with these PCTs, I think Some of them I imagine to be very specific. In other words, if they align to a certain region of the protein and can really just kind of hold it in place while it's being threaded through and folded into the 3D structure, that's likely to be very specific. There may be chaperone therapies where, even though we might not think of them as classic chaperones, maybe all they would do in the cell is to just simply slow down translation a little bit, right? The ability of these threads to get made. And sometimes it's really a matter of if the thread just gets spun out too quickly, then the thing just doesn't have enough time, and the cell just wants a little bit more time to make something that's functional. And maybe a line of chaperones, which would consider something that would maybe slow down that rate, therapeutically might be a lot more of a general solution.
And more applicable across
Absolutely.
various different proteins.
Yeah, does that make sense?
Yeah, for sure. Yeah. Okay.
Yeah.
Do we have any other questions? Okay. All right, well, thank you so much, Shaun and Wes, for your great discussions and insights. At this time, we'll ask you guys to jump on to your mobile apps and take a brief survey, and go ahead and enjoy the networking break that is going to follow after this. We want to thank Spark Therapeutics that was sponsoring this talk, and our next breakout session will be at 11:30. Thanks for attending, and we'll give one last round of applause for Shaun and Wes.